I am so excited about this next speaker because, as I mentioned in my little foreshadowing moments ago, um, Dr. Randall Kaye is going to share with you about a trial and an exciting molecule. And I have had the privilege of getting to know Dr. Kaye. I think I heard him speak at a Dravet meeting 3 years ago. My God. And he said basket trial. And I don't think I knocked him over, but I kind of tackled him. And I was like, Syngap, I want you, you've got to understand. And he sort of smiled at me and said, don't worry, it's going to happen. And I can't believe it's been that long, but it's happening. It's happening. And I've had the privilege of getting to know Dr. Kaye. And Sophia, if you want to raise your hand in the back, I knew one of the ways I judge companies is by who they hire. And when they hired Sophia, I said, these people are smart. These people are hiring quality human beings. And they've also since then hired Issa DeWoody, who is another legend in this space. So I've already talked too much and I apologize. But if you want to come up here, Dr. Kaye, please give him your attention and think about if this one's for you. A basket trial is an incredible opportunity for Syngap to get on the map. It's getting real, guys. And with great respect and appreciation, Dr. Kaye.
Thank you. Good afternoon. Nice to see everyone. Lunch was yummy. Thank you. My name is Randall Kaye. I'm the Chief Medical Officer at Longboard, and I also happen to be a pediatrician and immunologist by training. I've been working in this particular field for about 3 years, which I know is a really short period of time when you think about the journey that you all have been involved. And I will tell you personally, I'm enthralled and infatuated by just the journey that we've been participating in these 3 short years. I want to share with you a little bit of a story of what we've been going through as a company and introduce you to our medication and maybe a potential study that your loved one could be involved in. If not, it's okay. But I would like you to maybe think a little bit about, if not this study, maybe there are other studies or other research that you may want to think about getting involved in. And there's a reason for that. I need a slide. Corporate people can't talk without slides. So I know you can't even see this, right? There's all these there's some black, some blue, and some green stuff, right? But you know it's a really impressive bar graph, and it's probably important, or I wouldn't be showing it. But it's the anti-seizure medications that have been developed over the last period of time. And on the left-hand side, you see the 1800s, and I think potassium bromide was that first one. And then phenobarbital invented in the 1800s, and well, that's kind of weird. That's still around and still available. And then nothing happened for decades. And then we enter into this period where something's called second generation, which is basically people were kind of observing things and thinking they might work against seizures and started to utilize them. But they were never used in younger people. They were always used in adults. And the only way it ever came to the pediatric population is they would say, well, it's about 180 pounds, the kid, so about 50 pounds. So let's just do some math, which is probably about the worst way you could think about how do you expand a new medication to younger children. And then, frankly, there was a really long pause. And then we started to figure out how to do this right. And so you'll see this third generation group of medications that is just astounding of the number of treatment options. But I'll tell you, from the few years that we've been involved, it's still not the right way to approach the rare pediatric epilepsies that are out there. So I'm going to explain to you why I don't think it's quite right, but I want to give you some hope because I think we're getting an idea on how to approach it a little bit differently. Mike mentioned this, and yeah, he did think it was kind of a crazy idea when I met him a few years ago and was talking about this basket study, which is just a fancy word for saying, why don't we study these developmental and epileptic encephalopathies and study them a little bit more broadly? Because if we go the traditional route, we'll never get to that one. We'll do the more common rare epilepsies. Um, and so, just a quick on the characteristics of a DE, early childhood onset, often severe refractory epileptic seizures, developmental plateauing or regression, and then electroencephalographic or EEG findings that are abnormal interictally in between seizures. The criteria for a D is fairly broad, which makes it in some ways quite convenient when we start to think about how to study these conditions. And where do Ds fall in this big hierarchy? It's a small number. And a small number when you're a drug developer is actually a problem because we have these regulatory authorities that are looking for you to demonstrate that your medication is safe and efficacious. And when you have small populations, you don't have a large enough population to actually study, to have quote unquote statistically significant results and have confidence that your drug is truly going to be safe. So we have to approach it in very different ways. Well, if you look on the left-hand side of the screen, a bunch of companies that work in my therapeutic space have done it and done it pretty well. Treatment solutions for Dravet, LGS, CDKL5, TSC. There's probably 4 or 5 different approved therapies that are out there and that are really helpful and then actually make a nice difference. So as we start to think about our molecule, we ran into a roadblock, an ethical or scientific roadblock. We'll put them together, ethical and scientific. If we go this same route, then the vast majority of patients that are living with developmental and epileptic encephalopathies are never going to get a drug that's studied. And it's going to look just like that graph that I showed you in the very beginning. All these new medications, but we don't have a clue whether it can be applied safely and effectively into that population. You kind of see it on the right-hand side. So I don't know why I put this one here. This is my old slide. This is my old slide that has, there's probably 20 more DEs. That number is totally wrong. If you consider the genetic DEs, I probably should have a 500 or 600 in there. And I think every day that goes by, we probably discover more genetic DEs. So that number is vast, which again makes it very hard for us on the development side to prove to FDA that Syngap by itself in a treatment, a symptomatic treatment, is going to be effective if we can only enroll a relatively small number of patients. So I'm going to tell you a little bit about hope. I think this is a start. Things are going to change dramatically over the next 10 years. The molecule that we developed is a precedent molecule, meaning we looked at what other people were doing and we copied them. And I'll help you understand that in a little bit. We took the winnings and the learnings, but this is what I call as my baby. This is what my baby looks like. But I have a better picture. But it is a 5-HT2C receptor agonist. And it's entering into phase 3, and the goal is to treat a broad range of DEs. So, Mike, we don't say basket anymore. Now we say broad. I don't know why. It's what our commercial people tell me. I don't think it really matters, but broad range of DEs. That purple blobby thing, I get yelled at for saying this, but that's what it looks like in reality, but I'm not sure that's true, but that is what it looks like. And it binds into a 5-HT2C receptor. So we're pretty smart, but we're not that smart. There was a precedent compound called fenfluramine that you probably heard of, or Fintepla. Fintepla interacts the exact same way. It's a 5-HT2C receptor agonist, but it also interacts with a whole bunch of other receptors. So our thinking was, why don't we develop a molecule that only interacts with the receptor that you think contributes to efficacy? And if you don't know what it does, then don't let your molecule interact with it. And if you know it might cause harm or side effects or adverse events, don't let it interact with those receptors either. And that's all ours does. It interacts with that one receptor. So our predominant focus clearly would be on efficacy. So this is the name of our program. It's the DEEP Global Phase 3 program. I love having commercial people in my organization because we have all these like really cool names, right? So our company's called Longboard, and that's because our CEO is a surfer. I'm not, in case you haven't noticed. But we really love the sort of the ocean theme and the wave thing. We really do. So deep ocean is the study I'm going to tell you a little bit about, which is studying developmental and epileptic encephalopathies broadly. Deep sea is a slightly smaller study that will be focused purely on Dravet syndrome. Dravet syndrome is a fairly homogeneous, similar condition with a single point mutation. So we're going to study that condition separately. Our plan is to look and see if our compound works well across the spectrum from a homogeneous condition such as Dravet into our ocean study and studying DEs much more broadly. And then they kind of ran out of gas. So they got deep OLE, which I thought was OLE, but it's not. It's open label extension study. So one of the challenges that I think people are have when they start to even think about being involved in research is, are you kidding me? Am I going to roll my child in a placebo-controlled study? I hate that idea. But there's no other way that we can prove whether a drug is safe and efficacious unless we run a placebo-controlled study. That portion of the study typically is a couple of months. And at the end of that, we allow patients and families, if they're interested, they'll get access to drug on an open label basis. This is designed as a 1-year study. The last one that we did, we rolled patients into our open label study that went for a year, and we kept it going. That's what most companies do, is they allow patients to continue in the open label portion of a study until you have a definitive answer on that drug, and hopefully that medication gets approved. I'm going to flip through a little bit. I want to just give you a feel for what it might be like when you're involved in a study. I heard the conversation earlier about repurposing, which I think is a really neat approach. I don't think our taking a compound and maybe bettering a previous one is that much fancier than repurposing, because I think we've tried an elegant approach to taking a good idea and maybe making it a little bit better. We'll see what the data shows. But our approach differs in that we think it's important to have that safety and efficacy information so that we can go to FDA, so we can get it approved, and more importantly, so that insurance companies will cover the cost of it, whether they be private or public insurers. I'm going to focus now just on the 301 study. We kind of divide it into two portions. It doesn't matter at a patient level coming in, but we see the half the group that is in the study have DEs, a wide variety. We're not being that limited at all. You can have a gain of function mutation, a loss of function mutation. You can have any of the 500 or so genetic DEs. You can have TSC, you can have Syngap, you can have almost anyone as long as you meet the criteria that are listed here on the left. On the right-hand side is one particular group, which is Lennox-Gastaut. We expect that about half of the patients involved in the study will have LGS. Our plan is to do a comparison, because what FDA will say is, gosh, you ran this study and you only have 4 patients in there with, I'm making up numbers, 4 patients with Syngap. I hope more. You only have 3 patients with SCN8A, SCN2A. How are you going to generalize that to the hundreds and hundreds? And the way we're going to do that is we're going to compare the treatment, safety, and efficacy results between those two groups, and confirm what we saw in our original study that we provided top-line results in the earlier part of this year. I'm putting this up to slightly scare you and then slightly make you feel better. This is what it looks like to be involved in a clinical study. This is what your families will go through. There's a screening visit. You have to fill out a whole bunch of forms. You have to have an unknown for about a whole month to know whether or not your loved one is going to qualify. And then they go, after they meet qualifications, they are randomized to receive drug or placebo, and we gradually titrate up to the highest dose based on tolerability. There are 3 visits. Some are virtual. Some you physically go to the physician's office, and then we have the maintenance period. You stay on that exact dose for 3 months. You have a couple of interim visits where you're going in and you're having assessments. Usually they're questionnaires and forms to fill out. And each and every day you get to fill out and have the fun of a seizure diary. Now, some of you, I know you do this every day anyway. Some of you don't. It is a requirement of the study because that's the primary endpoint. That's the main thing that we'll be looking at. And then at the end of the study, you can say, that's great. That was fun. But we think we're done. In our last study, all of the patients that completed the randomized portion of the study opted to go into the open label extension. That's what we would hope we would sort of see. And then we continue to see patients on a regular basis, all at your primary's office or your specialist, and we follow you out as long as you're willing to keep us engaged. It gives more data. More data is good. So why in the world would you want to be involved in something that's so hard? You get a lot more time with your clinicians. You really do. The clinicians that we've chosen, we just came from our investigator meeting in Newport Beach yesterday, and they are some of the 30 or 32 top epileptologists that are in the country that are on the cutting edge. So it gives you access to a newer medication. And I think just a little bit of extra time is generally probably a good thing. I can't tell you whether your loved one's going to have a benefit from the study. I really don't know. I know right now, I think we have a good shot at showing solid safety and efficacy. But I do know that we'll learn, and we'll learn a tremendous amount of information from conducting the study. So that graph that I showed you before, where it kind of went like that, and it went like that, and then it kind of went flat, and then it went up. Even all those ones that are up there, they're not quite customized and thinking about specifically about your particular child or loved one. And I think this allows us to learn a little bit more. So I often use this quote, which is, consider research being part of your treatment paradigm. And if you do that, the more we learn, the more new medications we're gonna have. And I think that's the stuff that can give us hope as we start to think about the future. So I'm not going to show you this slide because it's boring. But I will tell you that we appreciate the partnerships that we've been developing over the years. As I started off saying, I know we're really early in this journey, and many of you are, shall we say, well into the journey with a loved one with Syngap, and let's try to be a part of it and see if we can come up with a treatment solution. So I'll stop with that. I have no idea if I went over my time, Michael, but if there is time for some questions, I'd love to.
We have about 10 minutes for questions. I just want to clarify. So for deep sea and deep ocean, it's placebo controlled. So 50-50, you'll get drug or placebo. But after that, you get to go into deep hole or OLE. And
did you say deep hole? I'm going to change.
Your marketing people should have really cut that. But that's open label. So everyone's going to get drug in that, right?
Everybody gets drug.
So I want to just underscore that because I have a habit of mansplaining things. And then the other question I have for you is, this is phase 3. So that means you've already done phase 1 and 2. And are you able to tell us a little bit about what you saw there?
I am. So we ran a study called Pacific. We released top-line data. We ran it in 50 patients. We did a 4-to-1 ratio, so 4 patients received drug and only 1 received placebo. It's referred to as phase 1b/2a study, but most people call it phase 2. And what did we see? We studied patients with DEs, and we had 3 groups that we were able to look at. Dravet, there were 3 or 4 patients in that group. LGS, there were 17 or 18 patients in that group. And then the D broad bucket, where it was onesies and twosies, a broad variety of different Ds. And then we looked at the treatment effect. and we saw a profound decrease in seizures. And it was really quite striking. And then we looked across the individual Ds, and we looked at Dravet, and we saw percent reductions in the 70% range, which is a lot. These are patients that were already on 3 or 4 anti-seizure medications. So these were the fairly treatment recalcitrant, treatment refractory patients. And in patients with LGS and the broad D group, we saw well over a 50% reduction in seizure frequency. So before you ask, I'll tell you. What else did we see? Because sometimes seizures isn't the biggest problem that you're facing. It was a short study. The primary endpoint was seizure reduction, looking at things like cognitive impairment, development, sleep, fatigues, all the other aspects that I know, frankly, are sometimes equally, if more important. It's very, very hard to look at that in a short-term study. It's a lot. This was an adolescent and adult study, so it's a lot to ask a drug to do in a month or two to have an impact on non-seizure outcomes if you've only changed the seizure frequency for a relatively brief period of time. But next year, which I think is just a month from now, we're going to release our top-line data for our open label extension study where patients have been exposed and on drug for a year. And that's where we can start to look at potentially some non-seizure outcomes. This study, Deep Ocean, Deep Sea, Deep Hole, I got to bring that one back. Or maybe not. Sorry. We will be looking at a variety of non-seizure outcomes. Looking at clinical global improvement, I think, is helpful, but there's other areas we'll be able to take a closer look at.
I'm gonna pontificate a little bit more, guys. I'm sorry. And then we have to Lindsay, who's I hope online. We have 10 minutes until our next group of speakers come because they're all remote. So again, I'm going to be dogmatic.
Yep.
Thank you. Sites. On that slide, it says there's 80 sites across the US, Australia, Europe, and potential regions. Two-part question. First part, because we have colleagues in the room from Austria, from Australia, from Argentina. What'd I miss? Probably some Canadians. This is a global study, right? So our colleagues from various countries, unlike an experience they often have a chance of participating in this. Maybe Portugal. We also have friends here from Portugal. But in the US, how does sites work? If I am in Texas and I'm already going to CHOP for my natural history study with SRF, or I'm going to Colorado, I'm going to Stanford, do I have to go to Texas Children's because I'm in Texas, or am I able to optimize that?
So it is a global study. US initiating the studies as we speak. They will continue to be initiated between now and probably March or April of next year, and that's just getting them started. It's probably about 30, maybe more just in the US alone. And then Australia, that'll be starting off in the sort of February, March timeframe. And then we will move into Europe. That will probably be in the early part of is April early part of the summer? And I think that's still spring. In mid-spring, we'll be going into Europe. There's additional regions that we're trying to get into: South America, Mexico, Canada, and some other regions that include different parts of Asia, but they're not official or finalized yet. So the two best things to do, maybe three: ask your doctor, ask them about their if your primary epileptologist is not involved in the study, they can readily refer over to a study center. The second thing you can do is, I don't know how to do this, but Sophia promised me this is going to work. Well, you can just call Sophia. Watch this. Ha, there you go. There's your email. Now you can go bother Sophia. Or you can click on that little QR code if any of you guys can figure out how to do that. Or you can go on clinicaltrials.gov. We update it every month. That tells you exactly what sites are involved in the study.
I want to make one more. Thank you. I want to make one more. I've got people are now whispering questions in my ear, and I see our next speakers are coming. So we have 7 minutes till the next speaker. Question one, how are existing meds handled? If you're already on meds, do you have to change that? Question from the audience. Second question, the inclusion exclusion slide, and I'll just say this because I've already been through this with you guys, is long and daunting. But the way I distill it is it's ultimately going to be, if you think you are qualified, you should call them. You should go to the site. You should let their world-class clinicians, and they really have chosen some of the best clinicians to be involved in this. But at the end of the day, it's countable motor seizures, right? To get this drug approved for our populations, we need people to go through the trial who have countable motor seizures. You've heard me say this before. Many of us, thank God, do not have countable motor seizures. We have obstinate seizures and we are well controlled. Please correct me, Dr. Kaye, if I'm wrong here, but if you do not have visible motor seizures, you're unlikely to get included in this. But if you do have countable motor seizures, this is very exciting. And the specificity, Dr. Kaye mentioned fenfluramine, that is a very powerful anti-seizure drug that some of our friends of Dravet are on. But the off-target effects are really unpleasant. What they've done with this drug is they've gotten rid of the parts that have the off-target effects, and they've just kept the magic that stops the seizure. So if you have a child who has countable motor seizures, there is a good chance you will be included in this, and you should be really excited about this. So those are 2 questions to you. Is what I just said correct, and how are existing meds handled?
It's correct. We don't change the medications patients are on. We won't even change them during the study if at all possible. We want to see how well our drug does on top of what would be referred to as standard of care. Patients could be on up to 3 or 4 anti-seizure medications. They can continue to take their rescue medication, can be in a ketogenic diet, can be receiving VNS. Don't make any changes. So we make that part pretty easy. Your second part was around the primary endpoint. You do have to have a minimum number of countable motor seizures. It's not a hugely high number unless your loved one doesn't have any at all, but it's 4 a month. That's the number that we're looking for on a fairly regular basis. But one correction, we still review and enter into the seizure diary every seizure a patient has. So if your loved one does has absent seizures or focal motor or others, they're still captured. So we're going to be able to analyze it. It's just from a regulatory standpoint. Unfortunately, countable motor seizures is the easiest, most objective, and most simple and straightforward one to do, and that's what FDA looks for. But we will count all of them, and we will report that back.
Thank you. Okay, Allison.
Side effect profile, the side effects that you've seen?
So the initial study was conducted in adults and adolescents. The most common adverse events were somnolence, CNS effects, and GI. And so you can imagine in the D population, it requires a little bit of interpretation from the parent. So a child that's pushing away food or doing food refusal is probably a GI side effect. And a child that is not acting like themselves might be sedated or somnolent. But those are the 2 categories that we predominantly saw. The vast majority of adverse events occurred within the first couple of days of dosing. Remember that titration period that I showed you? While patients are going up on drug is most typical. What we found once they were in maintenance period, we really didn't see a lot of new emerging unusual adverse events in our open label extension study. Nothing new emerged, but it's predominantly GI and CNS. But we've modified our titration period a little bit to go a little more slowly. I'm sure you've noticed when your epileptologist, neurologist puts you on a new medication, they go pretty slow. 2 weeks, 3 weeks, 4 weeks, and then they go to the next step. In a clinical trial, we have to go a little bit faster. So we go, originally, our original study was every 5 days. We go every 7 days. But during the study, you can go up and down. So if in the study not tolerating, not like themselves, food refusal, and go back down to the next one that they were tolerating well. And then wherever they land at the end of the titration period, that's where you go into the study. Thanks for asking that question. Yes.
Oh, sorry.
Part. So, okay, would you recommend patients where fenfluramine was not effective to take part in your study?
It's a very hard question. I'm going to repeat it. The question is, what about patients that were on fenfluramine and let's just say it wasn't effective or there was bad tolerability? That patient is excluded from our study. Mechanistically, our drug and fenfluramine works the same way. So for this pivotal phase 3 trial, we made the decision to not include patients that did not have a good experience with fenfluramine. So it was framed as a failure. The only reconsideration that we've had around this is if someone was on fenfluramine, but let's say they were doing fine, but didn't really want to do the echocardiograms or insurance wasn't covering the cost, but they didn't fail, we could consider having that patient come in. But unfortunately, by and large, a fenfluramine failure would be excluded. That's the subject of a study to be named some other day if we ever want to look at fenfluramine failures or do a switch study.
Thank you very much.